Professor Levitan, Rector of Tel Aviv University, Professor Makori, Dean of the Sackler Faculty of Medicine, our dear friend and guest of honor, Mr. Lee Corman. Perhaps I should address you as Dr. Corman, since you have just been awarded the well-deserved honorary doctorate of Tel Aviv University. Tel Aviv University faculty, our dedicated administrative staff, students and fellows, dear guests. More than 10 years ago, a group of biomedical scientists at Tel Aviv University recognized that without an SPF facility at the university, we would not be able to carry out cutting-edge biomedical research. SPF stands for Specific Pathogen-Free. 3 words that you will hear a lot of time today. The scientific jargon for the environment in which laboratory animals are maintained under strictly sterile pathogen-free conditions. Such an environment is absolutely critical for breeding and growing animals that are suitable for medical research, particularly those on which we model human diseases. Indeed, the official name of our new facility is the David and Inez Myers Building for Transgenic Modeling of Human Disease. The vision for an SPF facility was there, and as you will see later in the presentation, we also had superb engineering and construction teams to carry out the mission. The biggest challenge was, however, to raise funds to build such a sophisticated and expensive facility. From the very beginning, we received tremendous support from the university. I'm talking not only about our colleagues and deans of the Sackler Faculty of Medicine and the George Wise Faculty of Life Sciences, but also the leaders of the university. And here special gratitude goes to Professor Shimon Yankelevich, who as Vice Rector at the time immediately and wholeheartedly supported the idea. Professor Yankelevich is a physicist, but he needed very little convincing to recognize the vital need for this biomedical facility. Later, as rector and then pro-rector, in fact to this very day, he has worked tirelessly to help us move forward, to overcome obstacles, and to raise funds to outfit the building and equip the laboratories. There couldn't be a more appropriate moment to thank him. Special thanks go also to our Vice Dean, Professor Ruchalgi, who worked with us throughout this process in our previous capacity as Vice President for Research of the University and now as Vice Dean of the Sackler Faculty of Medicine. But the initial challenge was to get the big money for the construction of the building itself. We needed a major donation that would allow us to carry out the planning and begin the construction. And this is where our guest of honor, Mr. Lee Corman, stepped in. To those of you who don't know Lee, and perhaps by now there aren't too many people at Tel Aviv University in that category, Lee is one of the leaders of the Jewish community in Cleveland, Ohio, a major philanthropist and a director of the David and Inez Myers Foundation. Under his leadership, the foundation has become a leading supporter of science and education in Israel, with activities ranging from funding students in the Jewish Ethiopian community to advancing scientific research at our universities. At Tel Aviv University, the Myers Foundation established the Myers Laboratory and Chair for Cancer Genetics, and today we're inaugurating the Myers Building for Transgenic Modeling of Human Disease. Lee, I'm sure this is a great moment for you, as it is for us. Additional general contributions, no less important, were made towards completing the construction and purchasing the precious equipment. You will see the names on the honor wall, but I want to extend our gratitude to each and every one of them. Our Argentina friends of Tel Aviv University, the Kipp family, the Horace Goldsmith Foundation, and Ms. Susie Steinling of Vienna, Austria. Without this contribution, we would have not been here today. This building would have not risen without the superb design and excellent work of the university architect, Mr. Yoram Eldan, and the extremely professional and dedicated team of the engineering and maintenance division of the university, headed by Mr. Ofer Lugassi. The facility that we are inaugurating today represents a major achievement of engineering and construction. It had to be built on a small parcel of land, which necessitated expanding vertically rather than horizontally. Mr. Eldan and the engineers came up with excellent and original solutions to overcome this constraint. Special thanks also to engineer Shaul Agatstein, who specializes in animal facilities, who designed the interior of this facility. We're greatly indebted to the staff at the existing animal facility, the former director, Dr. Naam Kariv, and the new director, Dr. Miki Harlev. Dr. Harlev will head and operate the new facility, a job he is already performing at the highest professional level. And finally, the consumers, my fellow scientists, students, and research fellows. They're all deeply indebted to the university for taking up the challenge to establish this facility in these difficult times for higher education in Israel. In fact, this event is a reminder to our government that without fostering scientific research at the research universities in Israel, among others by helping them to build the requisite infrastructure, Israel will lose its edge in modern science with extremely grave consequences for the future of our country. 2 days ago, we heard some encouraging promises from our education minister, Mr. Saar. Let's hope that they are fully fulfilled. I'd like to conclude by thanking again all of you and inviting our rector, Professor Dani Leviatan, to the podium.
Thank you, Yossi. Dr. Lee Korman, President of the David and Inez Myers Foundation, Professor Yossi Mekori, Dean of the Sackler Faculty of Medicine, Professor Yossi Shiloh, Incumbent of the David and Inez Myers Chair in Cancer Research, faculty members, ladies and gentlemen. Actually, all that I have to say was covered by Yossi, but nevertheless I will repeat shortly a few things. I'm delighted to be here for the inauguration of the David and Inez Myers building for transgenic modeling of human disease, as we call it, the SPF, specific pathogen-free building. As you know from Yossi Shiloh's chair, this is by no means the first donation Tel Aviv University received from the Myers Foundation. In fact, there is also the Myers Lab for Genetic Research, and here, the Dean of Life Sciences, Professor Yoel Klug. Welcome. So there is the Myers Lab for Genetic Research and the Student Loan Fund. We are grateful to Mr. Korman for his generosity. Mr. Korman was appointed by the Myers to use the fund of the David and Inez Myers Foundation, which he had helped them create to benefit humanity. And I believe that his decision to help us in this endeavor, as in the previous ones, indeed serves well their wishes. I won't mention all of Dr. Korman's activities helping Israel. Suffices to say that he supports projects from Kiryat Shmona to Dimona, including in several universities, not only Tel Aviv University. So we are very glad to have Dr. Korman with us, and I have to present to you, Dr. Korman, the certificate, and so please come forward, and let me first read it to everybody. This is to witness that on the 10th day of May 2010, the David and Inez Myers building of transgenic modeling of human disease was inaugurated in the presence of Dr. Honoris Causa S. Lee Korman, President, David and Inez Myers Foundation, thereby providing a state-of-the-art facility for conducting genetic research at Tel Aviv University and throughout Israel, promoting technologies essential to modeling genetic disorders, determining gene functions, and evaluating novel drugs, supporting research that will lead to the alleviation of suffering and the saving of human lives, and for enhancing Israel's scientific stature and contribution to biomedical research and the medical progress. Signed, Professor Yosef Klafter, President of Tel Aviv University.
Yes.
So,
Thank you.
Let's come into the picture. Yeah, it's okay? Okay.
Thank you.
So
Now it's your turn.
now it's your turn Lee,
to address
to address this distinguished audience.
As a former kibbutznik, I have the prerogative of not wearing a tie at this formal event.
Yoel is not wearing it either.
Right. Yes. Dean, Rector, distinguished faculty, and friends, thank you for granting me a few moments to make 3 brief statements. The first is personal, as I want to say publicly what I've already said privately, that my being awarded a doctorate on Saturday evening has more to do with the reputation of my sponsor, Professor Yossi Shiloh, and the quality of his laboratory and his people than the merits of the recipient. Yossi, I'll cherish my degree, entwined as it is with our enduring strands of friendship, forever.
Thank you.
My last 2 statements are on behalf of the David and Inez Myers Foundation. So I would like a fellow trustee of that foundation, Bobby Goldberg, to come up with me. He, as you know, is the chair of your Board of Governors. Right. Right.
It's okay. It's a hot day today.
Right. A schlepper. But a Zionist like no other. It's most appropriate that Bob stand with me because in his entire adult life, Bobby has been an uncompromising advocate for one of the goals of the Myers Foundation: truth, transparency, and unqualified ethics in the governance of public institutions. Thank you. Firstly, our foundation wishes to express its thanks to all those who conceived, designed, and built this facility. For reasons I'll state in a moment, your work is of tremendous importance to us, and the manner in which you performed with difficult challenges, both intrinsic and those beyond your control, has been remarkable, and we thank you. Finally, the Myers Foundation would like to answer its own question. Why are we here, and why are we here now? Regrettably, I'm not a great student of the Jewish canon, and I've never noted that my great friend Bobby is either. Nonetheless, I'd like to, I wrote this out, but I'd like to devote the balance of my, the balance of my remarks from the coincidence of Shavuot being only 10 days from now. And I'd like to borrow from an article in the Journal of the Hartman Institute in Jerusalem, actually written by a Tel Aviv professor and chair of your Department of Philosophy, Menachem Fish. Professor Fish reminds us that Shavuot, later transformed into a commemoration of Matan Torah, giving of the law at Mount Sinai, with the purpose of reminding us how our religion was formed, was in actuality the mandate to the newly freed people to be a national festival. Shavuot celebrates the establishment of sovereignty, and Fish quotes familiar passages from Deuteronomy to support that statement, that indeed the mandate with respect to Shavuot is indeed Israel's first declaration of independence. And like the American Declaration of 1776 and the current Israeli Declaration of 1948, it stated what kind of a nation Israel is to be. And therein lies the answer to my question of why is the Myers Foundation here and here now? Our foundation is especially interested in basic scientific research, attracted by its search for truth in the natural world. Our foundation is expressly concerned with Israel. And our identity, can our identity be more ardent, can we identify a more ardent Zionist than my friend Bobby. So that would seem to be the answer. We are at an Israeli institution that does great work in basic scientific research. One need only to see the work of Yossi Shiloh's lab. But the answer is more than that, and it is in the language of Shavuot, the message of Shavuot. For good or bad, a disproportionate burden of creating the kind of nation described in Deuteronomy, a nation that is inclusive, fair to all, compassionate, a nation whose ethics are not relative, is born in this land by its great universities, by Tel Aviv University. For Israel to be a morally and physically strong nation, a nation that will sustain its children and the idealistically oriented children of the diaspora, my kids, Bobby's kids, the universities of Israel, this university must be strong. And this place, here and now, where truth, unmitigated by relativism or compromise, where that truth is sought, defended, and disseminated, is where the Myers Foundation should be, as should be all people of goodwill. The Myers Foundation is indebted to you, all of you, at this university, for what you give us, and for granting us the opportunity to link our name with yours. The answer to my question is that Bobby and I simply could not be any other place than standing with you here and now. Am Yisrael Chai.
I'd like to invite now Professor McCory, Dean of the Sackler Faculty of Medicine, to address us.
Members of the Presidium, members of the board, dear friends and colleagues. I'm standing here today not only as a dean but also as an academic physician. Thrilled and excited. Thrilled just by thinking what a powerful experimental tool is now being added to our armamentarium of investigative means here at this faculty. Excited to the fact that this addition, significant addition, is being added and puts us as a leading cutting-edge research institution in this country and worldwide. So definitely what I have to do, and I was the lucky one to be the dean that we inaugurate today this new facility, is the fact that just to hold my breath and to see what are going to be the results and what is going to be the scientific yield, I'm sure it's going to be outstanding. So on behalf of myself and the Sackler Faculty of Medicine, I would like to convey my gratitude, first of all, to the initiators, Yossi Shiloh, Dr. Lee Corman, the Myers Foundation, the university administration, my vice dean, Ruti Shalgi, who was very active during at least the last stage, as far as I know. So, and I'm sorry if I've missed anybody, so definitely to convey my gratitude to everybody of those who have taken part in this scientific important endeavor. Thank you very much, and let's look for many happy returns. Thank you.
The new facility will be used jointly by the staff of the Sackler Faculty of Medicine and the George Wise Faculty of Life Sciences, and I'd like to invite Professor Klug, Dean of the Faculty of Life Sciences, to speak to us.
Professor Leviathan, Dr. Corman, the Myers Foundation, I would like to thank you very much for the many efforts and donations, and to add to this, friends of Tel Aviv University in Argentina who also contributed, and we are still not there, but we are almost, and we will start to work soon. I would like to to let the audience know that this project that Yossi has raised, I guess, started to raise 10 years ago or so, at least, was very, very difficult to raise. And that's why it took so long. We are the last university in Israel, I think, that does not have the SPF, and it's very, very important. We cannot do really biomed research without such a facility. There were lots of arguments, and there were 3 presidents in a row that worked with us, and all were supportive. And finally, we are there. Dr. Korman, again, I think this is really a big thing for us to start this. And we are very happy that we can do the work. And again, to Yossi for his 10 years work, but there are many 10s to go. Thank you very much.
I would like to make just one small comment. As I said, this was a group of scientists, and in fact, we worked together from the start. It was not only my own initiative. Everybody here recognized the need for the SPF right from the start. So I think that it is now time to move to our presentations, and let me just make sure that this works. I guess our computer went to sleep, but I'll try to wake it up as quickly as I can. And while this is happening, I'd like to introduce our first presenter. Actually, we have changed the order of presentations, and the first presentation will be made by Dr. Yechiel Alkabetz, and I will conclude with my presentation. Everything seemed to be working just half an hour ago, so I hope it will. I'll be able to revive this thing. Okay, here we go. And what we have here are 2 presentations that will actually tell you what it's all about, and show you actually what is the facility that we are inaugurating today. One problem is that we cannot host you in this facility. I think we lost connection. Okay, let's see. So this was, machines. Okay, I think I'll have to reboot it. Anyway, while this is coming up, hopefully, I'll tell you in a few words about Yechiel. Yeah, maybe you could help renew the communication between the computer and the projector. I think that one of the biggest joys that we have here at the university, when I say we, I mean old professors like me, is to see the young investigators who join the ranks of Tel Aviv University, establish their new lab. And Yechiel is a very good example for that. Actually, he got all his degrees from BSc up to PhD from Tel Aviv University, then went on to do his postdoc at the Memorial Sloan Kettering Cancer Center in New York, and just recently established his lab here. Thanks so much. And Yechiel is really working in an area which is at the forefront of biomedical research, the use of human embryonic stem cells in medical research to understand developmental processes and diseases. And he will actually explain to us, and maybe now I will really understand why we need the SPF. And then I will take you to a virtual tour of the facility. Why I cannot take you into the facility itself, you will understand from the PS talk.
Thanks a lot, Yossi. I'm really honored to be here. So good afternoon, distinguished guests. And so I will make a really short introductory words for the SPF. And when I talked to Yossi about thinking about like a little title for the talk, so we really thought about advancing the research in Tel Aviv University. But I think that we thought together, why not actually explain how can we both reach gold standards in animal welfare, and I think that one of the most important things in this really fascinating building is that you can really increase the welfare of animals, and by that advance our research. So, so maybe a few words just about the need of animals in biomedical research. It is well known that throughout history, scientists have been always using animals for biomedical research in order to understand how our body is working, and to develop new research strategies and techniques, finding cures and treatments for numerous diseases, and discovering, of course, new drugs, and once discovering them, testing them for efficacy and for safety in human beings. And of course, evaluating medical procedures before they are used on people. So I think that all of these, by all means, have been achieved by using animals in biomedical research. Many of the diseases, many of them that our ancestors actually experienced firsthand, have turned out to be completely unknown for our offsprings. And the reason for that is obviously that most of them, many of them, have been just simply eradicated.
Yeah, cannot be heard. Thanks. That simply these, many of these diseases have been eradicated, and many others that are still here, present, can be actually controlled. And again, all of this is due to findings from research in animals and biomedical research. So I think a very strong argument why we do need animals in biomedical research is that both humans and animals are benefiting from this research. For example, life-saving surgical procedures, both animals and human beings are benefiting from this. Cancer therapies, organ transplantations, safe consumer products, and vaccines, of course, and treatment and cures for numerous medical disorders. I think that it would be, everyone would agree with me that virtually every major medical advance for both humans and animals have been achieved through biomedical research using animals or animal models. And it's not that we're not trying something else. Of course, if we ask ourselves why we do not use other models, then of course we do. We do this and we use other methods whenever we can. For example, cell tissue cultures. We can take cells from animals or from humans and put them in a culture dish and actually investigate developmental events and diseases, of course. Biological processes in the test tube, biochemistry. They are all like long-standing, very old disciplines that are very important and they taught us very much about development and disease. And of course, cutting-edge computational models that are now in the genomic and post-genomic era are being really crossing disciplines together with biologists and medical researchers to try to understand very complicated processes. But I think that although all of these important complementary disciplines have been taught us a lot about biology and animal development and disease, we cannot really do without animal research in biomedical discipline, and because these methods simply cannot mimic the complexity of our bodies. So, and this is where animals actually comes. So the advantages. I think that we all know that we both share quite a bit of similarities with animals, or, for example, mice that are depicted in these pictures in many organs and many body systems. So we both are vulnerable to the same diseases. And the relatively short lifespan of these animals allows us actually to study them throughout their lifespan, which means that we can investigate longevity, cancer, and other development and disease. Eventually, we can control the living environment of these animals and minimize experimental variables. Of course, that when we do experiments, so researchers are doing experiments and repeating them again and again because they want to get statistically significant results. And also, when we do it with these little creatures, we do need an environment that would be very tightly regulated. So all of these are, of course, an advance to our research. And these are just like, I'm sure that it's a very short list and I'm not going to go all these, but these are all actually virtually were achieved by using animals in biomedical research. But maybe one word about actually the other side of the cone. So, actually, maybe before, I would like to just show you one example for modeling human disease in mice. Okay, so the genetic constitution of mice is very similar to us, that we all know. And using genetic manipulation, we can actually create a mice or a mouse a genetic disease model which is similar to a specific genetic disorder that we have in human. So once we can actually create such a mouse, okay, and it could be for any genetic disorder, for example, a metabolic disease like Tay-Sachs, or a genetic predisposition to cancer or chronic diseases, we can make this mouse model based on the human disease, and this will allow us to understand the molecular basis of disease, and later on to maybe even find new drugs and treatment modalities. But so the slide that I was referring before, and I forgot this one, was basically while seeking for ways to advancing our research, I think that we really need to think about the SPF as really treating the animals from the other side, about their welfare, and I'm talking about their welfare. So can we also find ways of increasing animal welfare while reaching our gold standards in research? And of course, that when you think about animals for research, population density, nutrition problems, temperature and humidity fluctuations, and noise, all of these are parameters that could really affect our experiments because they firstly affect the welfare of the animals. And if they are getting into stress and disease, so what we are reaching actually is a drawback in our research rather than advance. And this is why we really need a very, very healthy and tightly regulated and animals that are actually growing in a tightly regulated environment. So we actually we are giving one thing, but we are earning two back. We are giving a tight regulation and animal welfare, but we are getting advanced in our research. So our new SPF. So SPF stands, as Yossi said before, specific pathogen-free. I would only use one or two slides just to say something about the SPF before we go into the visit into the SPF with Yossi's presentation. But the idea of SPF is that SPF animals are designated to be free from specified pathogens. It doesn't mean that we know that these animals have no pathogen at all, but at least we have a very concrete list of the pathogens that are devoid of. And this is very, of course, important for our research. And for that, they really need to be raised in a biosecure facilities. Only if they are tightly regulated, we will know what is it that they are actually that they have been exposed to or they are carrying. Their health status needs to be monitored continuously and by very, very accurate diagnostic tools. The SPF status, basically, which stamps on each and every animal, provides us with an assurance or a certificate that the animals we use for research are not infected with any specific pathogens that are listed below, that are listed for each and every specific animals. So if you think about advantages, of course, I think I would conclude here that the SPF technique and the facility is extremely useful for basic and applied science research. And I would only give like one or two examples, of course, that everybody is aware of. Vaccine trials, we cannot think about trying a new vaccine on an animal if we don't know what are the viruses or the bacteria that these animals are exposed to. So if we are buying new animals that are SPF from a vendor and we're bringing them to a SPF facility, this is how actually we keep these animals pathogen-free. Same for immunological studies or even transplantation studies. Think about rejection of a transplant. We need to know exactly what is that the animal suffers from before we go ahead and transplant and look for the result. So everything is possible since the listed of interfering pathogens can be actually ruled out together with the purchasing or the growing these animals. So this is actually a picture of the fancy elevator in the SPF, and I would from here give the podium to Yossi to take us to the visit. Okay.
So, like I said, by now you probably understand why we cannot, unfortunately, host all of you in the new facility. But I didn't want to leave you just with the impression of the picture of the building and the fancy elevator. So I thought I would basically what I would do is take you for a virtual tour of the new facility. And okay, here we go. So this is what you will see from the outside when we unveil the building's name and then the honor wall. But what does it actually mean? What is in this nice tower just adjacent to the old animal facility? As Yechiel explained, we want to maintain a pathogen-free environment, which means simply that we have to have a strict barrier between the, if you want, dirty outside world and the clean area inside, in which there is a pathogen-free environment. How can we achieve this in our dirty world? Well, this is one of the hallways in the clean area. Looks like regular hallway, but you cannot just go inside this hallway just like this, like you were entering this building a few minutes ago. You'll have to go through a special room where you will wash yourselves, and the workers will actually take a shower. Visitors will just wash their hands very carefully, very much like entering an operation theater. And you'll have to wear everything that you have to wear in an operation theater, shoes covers, these coats, sterile coats, gloves, masks, and caps. And then, only then, you will be allowed to go into this elevator, which is actually part of the clean part of the building. And here we are in one of the rooms in which we maintain the animals. And I took a picture of the ceiling here because this is where the air comes from. How do we maintain the pathogen-free atmosphere? This is essentially the essence of this facility. You have to have in the entire clean part of the building, which is most of the building, 5 floors, completely almost sterile pathogen-free atmosphere, which means that the air cannot come just from the outside like the air we're breathing here. It comes through specific machines, and it's pumped into these rooms through filters. So once you're there, you're actually breathing sterile air. How can we achieve this in such a big building? Well, if you go on top of the building, this is the infrastructure which you will find there. These very sophisticated machines actually take care of all the, first of all, of the ventilation system in the building, this clean air, the filters and everything. This is quite an impressive array of very sophisticated machines. They are all up on top of the building. Now we have to keep the equipment sterile. Everything that goes into that environment has to be sterilized. So here is a huge autoclave. You put everything in one side, and then the other side is in the other room, the clean room. So you open that door, you get your equipment clean and sterile. But and here is the this is an important point. This is the steam generating machine. This is actually high steam pressure, and the building has its own systems, completely independent of the infrastructure of the university. So in case something happens in the campus, it doesn't matter. The building continues to operate smoothly, and this is one example. We get our steam from the central system of the university, but this facility has its own. So it has to have its own system for everything, and you can't imagine how many systems this building has to have independently of its own in order to be completely separated from the entire infrastructure of the university. What about things that you cannot put into an autoclave? This room with this frightening sign here, actually there are 2 doors here, and between them there is a small room which is a fumigation room. So equipment that cannot stand the autoclave, the heat and the steam, will be passed through this system. It will be fumigated, and then will be used inside the clean area. And of course, we have double doors throughout the building, separating in each and every floor the clean area from the dirty area. We have a clean elevator and a dirty elevator, so you can start to see the complexity of keeping and maintaining such sterile environment in a building like that. Now, the inhabitants. Most of the inhabitants will be these cute mice. This is the most important experimental organism as far as modeling of human disease is concerned, for various reasons that Yechiel mentioned. And I would like to mention an anecdote here. While we were submitting the, each and every building, new building in a university has to have the approval of the municipality of the city, Tel Aviv, the town hall of Tel Aviv, and it's the same for any building in the city, same for any construction here. And when we submitted the plans of the new building to the town hall bureaucrats, they got back to us and said, well, there is something very important missing here, which is a parking lot. For a building of this size, you need such and such parking slots. That we want to prove that. And it took us...
Is all merit and everything...
No, we didn't do that. But it took us a lot of time to convince the town hall bureaucrats that the inhabitants of this building don't need all that parking space. In fact, this entire facility will be operated by a handful of people. These are still wrapped. You see, these are the carts on which the cages of the mice will be standing, and this is what they will look like when we start operating the facility in about 2 months. And of course, again, these each and every cage gets its own supply of sterile air. So these animals, they live actually very comfortably in a completely quiet, sterile environment with a very strict regimen of light and dark, and of course very good food. So they really enjoy the luxurious conditions of a 5-star hotel. And we have laboratories there. We need to do a lot of procedures, but no less important is the research. And in fact, in order to model human diseases in these animals, we need this technology, which is either transgenic or a technology called gene targeting, in which we target a specific gene and create a genetic disease. And this is actually, in fact, our final aim here. We have the space, we have part of the equipment, we're not there yet, but in fact the technology is already here. The transgenic technology has been used in our department for several years. It was brought to our department by several members of our academic staff, like Shimon Efrat and Ruta Sheri Padan. And more recently, the gene targeting technology was introduced to our department by Dr. Yael Ziv of our lab. So we have the know-how, we have the technology, we have the space. We just need to equip these labs and also get to academic positions from the university in order to operate them. So this is still down the road, and I think that by this I'll conclude and invite you all for the unveiling of the building's name and the honor wall inside. Thank you very much. Thank you very much for coming. So we will just go now to the building for the unveiling.